Detection of urea in urine. Urea is present mainly in the urine of all mammals. Urea is naturally produced during the process of the breakdown of proteins by the liver. Due to this process, amino groups are removed from the amino acid present in the proteins. These amino groups are converted to ammonia, which is highly toxic, and ammonia is ultimately converted to urea. To detect urea in urine, URINE and sodium hypobromite tests are done. URINE test. Materials required: urine sample in a test tube, phenol red indicator, 2% sodium carbonate, 1% acetic acid, soya bean powder, spatula, and a dropper. Procedure: using a dropper, take some phenol red indicator. Add 4 to 5 drops of phenol red indicator to the test tube containing urine sample. Take a few drops of 2% sodium carbonate solution. Add sodium carbonate solution drop by drop until a pink color develops in the test tube. Take a few drops of 1% acetic acid using a dropper. Add acetic acid to the test tube drop by drop until the pink color disappears. Using a spatula, take some soya bean powder from the watch glass. Add soya bean powder to the test tube and shake it well. The pink or red color appearing in the solution indicates the presence of urea in urine. Conclusion: at optimum pH and temperature, urease enzyme present in soya bean powder decomposes urea into ammonia and carbon dioxide, which form ammonium carbonate. Ammonium carbonate is an alkaline substance which changes the slightly acidic solution to alkaline solution. Phenol red is used as an indicator in this reaction mixture, so the color of solution changes from yellow to pink. Detection of sugar in urine. Sugar is ordinarily not present in normal urine. When the sugar level in blood rises above normal level, the kidney eliminates the extra sugar through the urine. Sugar then starts to appear in urine. Generally, Benedict's and Felling's tests are used to detect the presence of sugar in urine. These tests give colored precipitates such as green, yellow, and brick red, depending on the concentration of sugar present in urine. Benedict's test. Materials required: urine sample in a test tube, Benedict's reagent, burner, test tube holder, and a dropper. Procedure: using a dropper, take a small quantity of Benedict's reagent. Now, add the reagent to the test tube containing urine sample. Boil the sample over a burner for 2 minutes, holding the test tube firmly with the test tube holder. Keep shaking the test tube as it is being heated. A brick red precipitate appears, indicating the presence of sugar in the urine. Conclusion: on boiling the urine sample with the Benedict's reagent, the cupric ion present in the Benedict's reagent is reduced by the reducing agent, sugar, to form a brick red colored precipitate of cuprous oxide. Therefore, sugar is present in the urine sample. Detection of albumin in urine. Albumin is a protein produced by the liver and is mainly found in the blood. A trace of protein is found in normal urine daily. Under pathological conditions like albuminuria, the level of albumin found in urine will be way above the normal level. In kidney disturbance and in high blood pressure, albumin level in urine is significantly high. To detect albumin in the urine, sulfosalicylic acid test and HELAS tests are done. Sulfosalicylic acid test. Materials required: urine sample in a test tube, 30% sulfosalicylic acid and a dropper. Procedure: using a dropper, take a small quantity of sulfosalicylic acid. Now, add it to the test tube containing the urine sample. A cloudy turbid solution or precipitate appearing in the solution indicates the presence of albumin in the urine sample. Conclusion: when sulfosalicylic acid reacts with albumin, it causes denaturation of protein in the sample that appears in the form of precipitate.
Hayes test for detection of bile salts. Hayes test is used to detect the presence of bile salts in urine, which may be any of the sodium salts of the bile acids such as taurocholate and glycocholate. Bile salts lower the surface tension of urine. When sulfur powder is added onto the surface of normal urine, they float. When bile salt is present in urine, it reduces the surface tension and the sulfur powder sinks to the bottom. Samples: freshly voided urine, preferably clean catch midstream morning specimen. Samples can be stored at 2 to 8 degrees centigrade for 24 hours. Grossly contaminated or unlabeled samples should be rejected. Reagents: sulfur powder. The sulfur powder must be absolutely dry and the particles small and of uniform size. Store the reagent at room temperature in a cool, dry, well-ventilated place. Equipment: test tubes of dimensions 15 by 125 millimeters. Spatula. Test tube rack. Procedure: all standard precautions should be followed. Mix the urine sample well. Take 10 milliliters of urine in a test tube and put it in the standing position in a test tube rack. Add or sprinkle dry sulfur powder on the surface gently and observe the particles. Results and interpretation: if sulfur particles sink to the bottom of the test tube, it indicates the presence of bile salts in the urine sample. If sulfur particles do not sink and float on the surface, it indicates the absence of bile salts.